and welcome to the course Social Behavior and the Brain, an introduction to social neuroscience. I am Dr. Ark Verma, an associate professor at the Department of Cognitive Science at IIT Kanpur. This is week 7. We will continue talking about various aspects of regulation of social behavior, and in this lecture specifically we will talk about asymmetric frontal activity in the regulation of behavior. We will take, for example, the case of valence and motivation and anger, and we will discuss this in some detail. Now, so far we have seen aspects of how prejudice of various forms gets activated instantaneously almost, if you remember the Ito and Erland studies, and once it is activated, it needs to be controlled through processes of regulation, either through automatic or through deliberative mechanisms. So, we have seen so far, starting from face perception, empathy, and even, for example, things like how racial discrimination etc. sets in. You are showing people faces of white participants, faces of black colored people, faces of white people. There are these instantaneous informations get activated. So, race, gender, age, all of these things get activated very quickly. If you remember the N100, N200, P300 components. Now, once all of this information is instantaneously available, it automatically starts affecting the mental processing of an individual for those given schemas. A lot of times it converts into prejudice. Also, it stays, it basically manifests as prejudice even if the person does not want to come out as prejudice. So, a lot of studies we have seen so far that people professed being egalitarian, they professed not wanting to affect that prejudice, not wanting to get affected by that prejudice in their decisions, but the prejudice still sorts of seeks through. Now, in that case, given that it's not socially appropriate to come out as racially biased or casteist or communal, people have always tried to regulate their urges or regulate their automatic tendencies for prejudice and discrimination. Those regulative mechanisms we have studied in detail, they operate through either automatic aspects of behavior or they operate through deliberative mechanisms. More recently, in the previous lecture, we have discussed the aspects of emotional decision making. We have looked at various theories of how emotions can basically complement the process of decision making. How can they be part of this process of decision making, contrary to what people have already thought, that emotions are detrimental to rational decision making. Not really that, because we have seen in the last few lectures that emotions can actually form a very important part of how decision making is done. We have also learned about mechanisms that allow for emotions to play a part in decision making and also allow for emotions to be kept away from decision making when they are not acting as relevant information. So, we have done so far. We have also in the most recent lectures looked at the neural basis of decision making. For example, we have looked at several theories, for example, the somatic marker theory or the dynamic filtering theory and so on. So, this is basically the recap so far, and now let us take this conversation forward. Now, we already know that one of the organizing principles for emotional information is valence, something being positive versus negative. Positive emotions are things like happiness and pleasure and joy and so on, whereas negative emotions are fear, sadness, disgust and so on. So, valence, the positivity or the negativity of emotions, has actually been used to organize experimental findings of the relationship between left versus right asymmetrical activations in the frontal cortex and that of emotional experience and expression. Now, here in this lecture and the lectures coming forward, I will introduce you to the idea of asymmetry or lateral asymmetry or hemispheric asymmetry. Now, so far you have seen me mentioning areas of the brain, basically the limbic areas and the frontal cortex areas, the medial prefrontal cortex, the dorsolateral prefrontal cortex, the anterior cingulate cortex. We have also talked about, say for example, the thalamus and the hypothalamus, the precuneus and so many other things. One of the very interesting aspects of the brain is that there are two hemispheres of the brain, the left hemisphere and the right hemisphere, and for certain tasks the left hemisphere is more adept, whereas for certain other tasks the right hemisphere may be more adept. It does not mean that at any point in time the left hemisphere exclusively performs a task or the right hemisphere exclusively performs a task. It just means that for that specific cognitive function, the left hemisphere may be contributing more to the overall decision making or to the overall processing. This particular phenomena can be described loosely as hemispheric asymmetry. Established hemispheric asymmetries are for cognitive functions like language production, wherein we know rather definitively that the Broca's area in the inferotemporal part of the left hemisphere basically is responsible for production of language. Also, for example, we know that the right hemisphere, the fusiform area in the right temporal lobe is responsible for perception of faces. Broadly put, left hemisphere is basically ascribed to doing functions such as language, mathematical functions and tool making, motor functions and so on, whereas right hemisphere is basically used in typically implicated in understanding figurative aspects of language, music, understanding visuospatial information and so on. Now, where emotions are concerned, the right hemisphere is supposed to be particularly active, particularly important for processing of emotions, but here what we are going to talk about is asymmetries in the frontal cortex. We are not talking about the asymmetries of the overall two hemispheres and how they function with respect to emotional processing. We will try and focus in the asymmetries in the frontal cortical activity. So, this lecture and the other 3-4 lectures in this week, I am going to be focusing on asymmetric frontal activity either towards the left side or towards the right side, and how does this happen, in response to what kind of stimuli it happens and so on. So, please keep an eye out for this. So, coming back, we were saying that valence has been used to organize experimental findings of the relationship between the left versus right asymmetrical activations in the frontal cortex, and that basically linked with both emotional experience and emotional expression. More specifically, what has been found is that positive effect has been related to greater left than right frontal cortical activity. So, it seems that the left frontal brain responds more to positive effect, positive emotions, positive valent ideas, whereas the negative effect has been found to relate to greater right than left frontal cortical activity. So, in the frontal cortex, your prefrontal cortex in the front part of the brain, the left part basically reacts to more positive information, whereas the right part reacts to more negative information, and this has consequences for processing, which is what we are going to understand as we go forward. So, this relationship has been sparked or has been discovered by systematic observations. For example, that damage to the left frontal cortex leads to depression because you are not able to process positive affect, positive information, positive emotions, whereas damage to the right frontal cortex causes mania because you are basically not able to mellow down any of the positive hysteria that comes through by sort of dampening it by processing negative information. Current research demonstrates that both trait and state positive effect was associated with increased left frontal cortical activity, whereas trait and state negative effect was found to be associated with increased right frontal activity. Now, at this point what you have to do is you have to treat the frontal lobe as a overall brain, and basically think of right frontal brain as the left frontal brain and how they are involved in the processing of negative and positive information respectively. Now, similar results like the one I just mentioned have also been obtained using a wide variety of neuroscientific methods, including things like lesion studies, repetitive transcranial magnetic stimulation, positron emission tomography, the PET scans, the event related potentials, the electroencephalographic data, and also these effects have been observed with both human studies as well as animal studies. So, this asymmetry in frontal cortical activity with respect to positive versus negative emotions has been replicated, has been found through different methods, and has been found in both human and animal subjects. Further, it has been proposed by Harmon Jones in Harmon Jones 2011 that studies till in the 1990s typically what they were doing is they were seeking to examine the relationship between asymmetrical frontal activity and emotion. That basically studies that sought to examine the relationship between asymmetrical frontal activity and emotion confounded affective valence with the direction of motivation. Now, it is very interesting that whenever we are encountered with positive emotion, there is also a tendency to approach those things. So, anything that we like, anything that inspires joy or happiness or elation, there is an automatic approach tendency towards it. All right. Similarly, anything that we don't like, say for example fear or disgust or any other negative emotion, like even anger for that matter, but we will talk about anger in more detail going forward. Anger for that matter, the aggression for that matter, typically leads to a withdrawal response. Now, what people have said, I mean the authors of this particular chapter, they basically point out is that till the 1990s, most of the studies that have looked at the study of emotional expression and emotional experience, they have rather confounded the typical valence part as well as the motivation direction of motivation part. So, while these things are very similar and they may be correlated, the author's point is that they are not identical and should not be confused as such. We should be able to tease apart the effects of valence and effects of approach, because it is possible that there are things which are not so positive but still invite approach motivation, and are not so negative but still avoid basically incite avoidance motivation. So, we should study that also. So, more specifically, what has happened in the literature so far is that all positive affective state or traits, for example joy or interest or curiosity, that have been empirically examined were typically examine approach motivations, whereas all the studied negative states and traits, for example fear or disgust, were typically confounded with withdrawal motivations. So, they point out a very interesting gap in the literature, and they basically point out the need to tease apart these two aspects, these two conceptual variables. How would we do that? Now, to understand whether these asymmetrical frontal cortical activations result from either affective valence or from motivational direction, the authors sought to examine a specific emotive state that avoided the confounding of valence and direction of motivation. So, they were looking for a candidate emotion that may be positive but still incites avoidance, or that may be negative but still incites some kind of an approach motivation. Let us look at what they chose. Now, to that effect, the authors began investigating the relationship of anger with asymmetrical frontal cortical activity, and it is because past social psychological and animal behavior research has suggested that anger, still being a negative emotion, but it evokes in a lot of cases approach motivational tendencies. So, anger sometimes leads to approach tendencies. For example, if you are very angry, sometimes you might be interested or inclined to approach or behave or act out aggressively. All right. So, in that case, anger is a negative emotion, or it has negative valence but carries approach tendency. So, it seems to be at a crossroads for valence and motivational direction. Now, they basically thought, the authors, when they chose anger as a candidate for exploration, they thought that if asymmetrical frontal activity relates to motivational direction, then anger should relate to greater left than right frontal activity, because remember left frontal activity was linked with positive as well as approach motivation also. So, because anger is associated with approach motivational direction. On the other hand, if asymmetrical frontal cortical activity would relate to affective valence, then anger should basically relate to greater right than left frontal activity, because anger is always associated with negative valence. So, this is why they are choosing anger as a candidate for exploration. Now, by investigating the relationship of anger with asymmetrical frontal cortical activity, the authors were in a position to gain a more complete understanding of the psychological and behavioral functions of asymmetrical frontal activity. Also, basic research on anger and its underlying neural systems can also provide us insights useful for understanding the relationship of how motivational direction and valence are sort of linked with each other. Most contemporary theories, however, of emotion operate on the assumption that positive effect is only related to approach motivation, whereas negative effects are related to withdrawal motivation. So, this similarity anyways is a well established, well practiced similarity, but here we have a chance of teasing these things apart. Let us see how it comes across. So, by focusing on anger, the authors shall be placed in a better position to understand how these two dimensions, that is dimension of valence and dimension of direction of motivation, are related to each other. Finally, by understanding the basic process involved in anger, the researchers all in the discipline shall also be better equipped to explain, predict and control anger whenever required. So, we will talk about their entire enterprise in some more detail, but before we go there, I thought it might be a good idea to just revise some of the methods that we are going to be using. Alright. Now, most research on asymmetrical frontal cortical activity and emotion has relied on the EEG paradigm, electroencephalography paradigm, more particularly the power in the alpha frequency band. So, you might remember from the previous courses that electroencephalograph basically measures the electrical activity in the brain, which is as an outcome of the neuronal and the electrical activity in the neurons. A bunch of neurons are active simultaneously, and the cumulative activity of those neurons is measured in these EEG bands. Now, as I said, the raw EEG signal is a complex waveform that can be decomposed using fast Fourier transforms, wherein several frequency bands can be extracted from the unprocessed EEG data. So, typically when you place the cap with the electrodes on a participant's head, there is a lot of electrical activity going on in the brain. You want to really understand or process a specific type of activity contingent to specific type of mental process. So, what can be typically done as a beginning is you use fast Fourier transform to separate out the different frequency bands and study a particular frequency band that is of interest to you. So, for example, here we are going to be talking about the alpha frequency band. When you are talking about REM sleep and non-REM sleep, there are other bands that are used in depression. There are other frequency bands that might be useful, and therefore the area of interest, or say for example the frequency band of interest, is actually studied in more detail. Now, what is this alpha frequency band? Alpha frequency is basically that one that ranges from 8 to 13 hertz. And previous research has suggested that the alpha power is inversely related to cortical activation using a variety of other measures of cortical activation as well, such as fMRI. So, what is alpha? What is basically this alpha frequency band? It is the range of frequencies that operate between 8 to 13 hertz periodically, and it is inversely related to the amount of cortical activation present. So, when there is high cortical activation, there will be lower alpha frequency range. Also, remember that the EEG alpha power is inversely correlated with PET and fMRI measures, because those measures actually are directly correlated or linearly correlated with the amount of neuronal activity that is observed. So, it might be assessing different aspects of brain activity, for example pre or post synaptic potentials. We will talk about this as we go ahead. Ultimately, if you remember, the both positron emission tomography and fMRI measures typically depend upon blood flow to brain areas recently involved in neuronal activity, although other changes might also affect fMRI, such as oxygen consumption and blood volume changes. Now, remember, we have talked about positron emission tomography. What happens in positron emission tomography? A radioactive isotope is injected in the blood, and the way it travels to the brain, and wherever the brain is basically being engaged, for example, it is engaged in processing a specific type of stimulus, the idea is that there will be larger amount of blood flow to that region, and you can basically detect the larger amount of blood flow to that specific region by tracing this particular isotope. So, that is positron emission tomography, just very loosely defined. Similarly, in fMRI, we basically use the Bolt signal, blood oxygen level dependent response, where basically again it is contingent on the amount of blood flow reaching, or the larger amount of blood flow reaching the area of the brain that is highly active, where basically the mental functions start using up the oxygen, neurons in that area become deoxygenated, and that is basically what is measured. So, both of these measures are actually depending upon, as I said, on blood flow to the brain areas, and they are in that sense different from how the EEG activity is actually measured. So, since both PET and fMRI measure blood flow rather than neuronal activity, these activations are not really happening in the real time with with neuronal activations. They are basically after effects of neuronal activations. okay, and therefore they can be seen as blood responses to blood responses to neuronal activation. So once neurons have started processing, they start using up more and more oxygen, therefore they need more oxygenated blood, and that is what is measured by PET and fMRI. The interesting part here is that the EEG method actually provides a different kind of processing advantage. For example, for PET and fMRI, there is a biological limit on the temporal resolution of these responses. It is such that even in the best measurement system, the peak blood flow response typically, which we measure and which we see in those charts, occurs around 6 to 9 seconds after stimulus onset. Okay, so it is something that happens later or after stimulus processing has happened, and in that sense it cannot give you how the stimulus is being processed in real time. Although there are suggestions and people are working out experimental methods that could allow for stimulus condition differences as early as 2 seconds, but at the moment these methods PET and fMRI don't have a very good temporal resolution. Okay, so the limitation of PET and fMRI measurements are biological, whereas the EEG measures are typically instantaneous since they are measuring electrical activations directly and they operate on a sub-milliseconds resolution. So in that sense you can see there is a difference between the merits of the PET fMRI methods and the EEG ERP methods. However, spatial resolution of the EEG, which is the ability to determine which specific areas of the brain generate the signal recorded, is not as good as the spatial resolution of PET and fMRI methods. See, both of these methods have a very important pro and a very important con. Whereas PET and fMRI have a very good spatial resolution, you can exactly know which area of the brain received that highest oxygenated blood and which area was basically involved in processing a particular stimulus. So you know the spatial region of the brain very well. Yes, the temporal resolution is pretty bad, so you don't know whether this region is doing the after processing or it's difficult to know whether this region was directly involved in processing of that stimulus, and that makes it difficult for interpreting the results from PET and fMRI. EEG on the other hand is directly measuring the neuronal activity that is there, but the idea is that the neuronal activity is a summation or accumulation of several neurons getting activated. Because it is a summation of several neurons getting activated and it is this activity is coming through the pia mater, dura mater in the skull of the brain, it is difficult to identify the source from which the specific event related potentials or the specific EEG frequency bands are coming from. So the spatial resolution of EEG is particularly bad. Obviously, a lot of work is being done to address this lacuna of this particular method as well, because people are working out mathematical resolutions to this problem, which eventually should allow for EEGs to have better spatial resolution. EEG research, on the other hand, if you compare the two things, is much less costly than fMRI or PET research. Remember, EEG research is typically non-invasive, whereas fMRI and PET research, PET at least is invasive, fMRI is non-invasive, but you have to go into the scanner, and sometimes people may have problems with the scanner, people complain of different kinds of fears and so on. Also it is very expensive, the maintenance and the purchase of an fMRI or a PET machine is very, very expensive, at least very expensive as compared to the EEG machine. Alright, so there is basically per subject scanning cost for an fMRI basically varies between 7 to 10,000 in an educational institution, whereas for EEG it is a very low cost, although there is a lot of cost of consumables when you are doing EEG. Alright, so there are, as I said, pros and cons in using the two methods. Finally, PET and EEG permit measurement of tonic, that is resting state or baseline activity, as well as phasic, that is when activity is happening in response to certain stimulation, whereas fMRI permits measurements of only phasic but not tonic activity. So this is again something, it depends on the kind of question you are asking, it depends upon what kind of brain activity you are mostly interested in. There is also a difference in terms of what kind of brain activity these methods are actually relying upon. Okay. So, this is just a bit of a primer about what kind of methods are typically used. We have done this earlier as well, but just to sort of revise this to you, because we are going to be talking about these methods as we go further. Now, scalp related electrical activity is the result of activity of the population of neurons. Large number of neurons are giving up these electrical activations, they are being summated, and what that in one electrode probably something that is happening over, let us say, 1 square centimeter, all of that is coming up. So, it is very difficult to disentangle exactly the source, the exact single cells, the exact neurons whose activity is getting recorded here. There are other ways, the invasive way of single cell recording, which will give you the exact idea of, okay, this particular neuron was responding to, let us say, a horizontal line or a vertical line, which people have done and typically do with animal studies, but not so much with human studies. Now, this activity gets recorded on the scalp surface because the tissue between the neurons and the scalp acts as a conductor, alright, voltage conductor. Now, since the activity generated by a single neuron is small, it is assumed that activity recorded by the scalp is an integrated activity of numerous neurons that are being active synchronously. So, they are all sort of acting at the same time, firing at the same time, if you want to use that. Moreover, for activity to be recorded at the scalp, the electrical fields generated by each neuron must be oriented in such a way that their effects cumulate. The electrical field should line up in such a way that their effects sort of get added up, alright. The neurons must be arranged therefore in an open field as opposed to a closed field sort of arrangement. What happens in an open field arrangement, the neurons dendrites are all oriented on one side of the structure, so that all the potentials are sort of getting summated, whereas axons are departing from all of the axons are departing from the other side. Now, open fields are certainly present when neurons are organized in layers, and this is best found in most of the cortex, parts of thalamus, the cerebellum and other structures. So, these are the regions when you are sort of talking about emotional computations, these kind of cognitive functions. These regions therefore are specially suited for measurement of EEG or ERP responses, because there is presence of open fields in these areas. Now, because of the need for the summation of electrical potentials, the EEG activity is therefore most likely a result of post-synaptic potentials, which typically have a slightly slower time course, and they are more likely to be synchronous over time and then summate than pre-synaptic potentials. So, again, these are all nitty-gritties about why a particular method is useful and why a particular method is useful for measuring specific type of cognitive function emerging from a specific region of the brain. This is all that I wanted to say. I will take this discussion forward in the next lecture. Thank you.